Hi everyone, my name is Alex, and for my project I've actually had the privilege of working with 3 different biology labs here in BU's biology department. And my project is on how the clownfish got its stripes. I investigated some of the mechanisms underlying this really unique coloration you see on the clownfish. And to understand my project, we have to learn a little bit about something called conspicuous coloration. And this is just the scientific name for really bright coloration in nature, and it's seen abundantly in the animal kingdom. And conspicuous coloration presents a puzzle for biologists because we would expect species that present bright coloration to be very vulnerable to attack from predators due to their high visibility. And we wouldn't expect these to exist, but they obviously do, which is why a lot of research has been done into organisms that present conspicuous coloration, especially in terrestrial systems. So because of this research, we now know that poison dart frogs actually convey that they're poisonous with their coloration, and certain bird species use coloration to distinguish between each other and to attract mates. Now, although a lot of research has been done in terrestrial systems, another group of highly colored organisms exist, and these would be coral reef fish. And there is actually a lack of research into why coloration occurs in coral reef fish. So the motivation behind my project was to start a preliminary conversation on why this coloration exists in coral reef fish. Now, for a senior project, looking at all coral reef fish would be too large a scope. So to narrow the scope down, we decided to look at one particular species, and that is the clownfish, specifically the Amphiprion percula. And overall, my research question ended up being which genes play a role in clownfish coloration. To kind of break down this overall research question, we took a 3-step approach, and I'm going to present that here in 3 questions. So the first step we wanted to take care of was to figure out what do we already know about clownfish coloration, and this basically involved a literature search to see what research was already out there and how we could incorporate these ideas into our project. The next step was to actually do an experiment and look and find which genes are different between colored tissue in the clownfish. A clownfish has orange, white, and black tissue, and we wanted to figure out which genes are being used differently in each of these tissues and therefore might contribute to this weird coloration we see. And this step flows into the final step, which is to figure out, okay, we know which genes are being used differently. Now, from a biological standpoint, what is the function of these genes? What are they doing in these fish? So I'm going to talk about the first step. What do we already know? And from the literature, we figured out things from 2 perspectives. First, we looked from an environmental perspective, and what we found out has to do with how clownfish live in these organisms called sea anemones. And sea anemones have stinging tentacles, which the clownfish live inside for protection. And we figured out that clownfish that live in an anemone with a weak sting actually tend to be that iconic orange color we're all used to. However, clownfish that live in anemones with a strong sting, such as this carpet anemone, tend to manifest something called a melanistic morph, which is a black color morph. So this was an interesting finding from the environmental perspective. The other perspective we looked at was from a genetic outlook. What you see on the board is a breeding chart from the aquarium industry, and what it tells us is that clownfish can be selectively bred to create really wacky colors. So you see a completely white clownfish up there, clownfish with lightning patterns, and the overall gist of what this breeding chart is telling us is that there is a hereditary component to clownfish coloration. In other words, there is a genetic basis, and this comforted us knowing that there was a genetic basis, so looking deeper into this wouldn't be a waste of time. So once we knew there was a genetic basis, we could actually go and look closer at that and figure out which genes are different in the clownfish, comparing the tissues together. And to do this, we carried out an experiment. We took white, orange, and black tissue from 9 clownfish, and we took these samples and put them in a fancy machine called the Illumina HiSeq 2000. The name isn't really important, but what it does is something called RNA sequencing. So all of our cells have the same set of DNA, but how it uses the DNA depends on the cell. Your skin cell is going to use a different amount of DNA than your liver cell per se. All cells have the same ingredients, but they use a different recipe. And so this recipe is presented in the form of a molecule called RNA, or the expressed DNA in the cell. And this machine tells us what types of RNA and in what amounts this RNA exists in each of our samples. And this data was given to us in the form of RNA libraries, which are digital files that we put into a software package called Trinity. Trinity organized the data and gave us an end result called a transcriptome. Like I mentioned, cells don't use their entire collection of DNA, and the transcriptome accounts for that by only looking at expressed DNA. So it's a lot like a genome, but it only takes into account genes the cells are using. Another way you can think of it is a snapshot of what a cell's genes are doing in any certain point in time. And this transcriptome let us kind of finish up and answer the question which genes are different. We used it in something called differential expression analysis, which sounds complicated, but what it did was pretty simple. It compared 2 tissues at a time for a total of 3 comparisons between tissues, and it determined which genes were over and underrepresented relative to other tissues. So that gave us the answer to which genes are different. We had those results digitized after this step, which meant we could go into the last and final step of what are these genes doing in the fish? What is their biological function? And to carry this out, we did another complicated sounding analysis called a gene ontology enrichment. But again, what it did was pretty simple. It basically took each of our samples and looked at the genes and then assigned biological names and definitions to each gene, which lets us know what each gene is doing from a biological standpoint in the fish. And what we found out was, first of all, for white and orange tissue, we weren't really able to find any conclusions there. The white tissue didn't have that many enriched genes, so we just couldn't draw any conclusion. And the orange tissue had an overrepresentation of genes having to do with muscle. So muscle development, muscle contraction, but there was no literature to back that up, so we didn't look into that further. However, the big takeaway from this actually had to do with the black tissue. And in the black tissue, we found that there was a large overabundance of overrepresented immune function genes. So genes that had to do with a cell's immune function, such as response to wounding, response to stress. And this was interesting to us because it kind of agreed with some pre-existing literature we found. And a lot of literature exists that shows that melanin, which is the pigment that creates colored tissue, colored black tissue, helps immunity. It helps insects fend off microorganisms. It helps humans and other vertebrates deal with wounding. And this is interesting because of the finding we found earlier, which is that clownfish that live in strong stinging anemones tend to be darker, and clownfish that live in weak stinging anemones tend to be orange. And this is just a figure from the literature that shows what I just said. The S. gigantea anemone, which is the strong stinging anemone, has a higher proportion of melanistic morphs living in it, while the weak stinging anemone has more. The weak stinging anemone, also called H. magnifica, has more orange clownfish living in it. And this is interesting because it suggests it's possible that the black tissue of the clownfish plays a role in preventing these clownfish from getting hurt by their host anemones and helping them with their immune function. So in the end, I'm going to go through those 3 steps that we talked about in the beginning. So in conclusion, how did we find out what we already knew about clownfish? We looked into their genes and their environment, and we found both of those things affect coloration in the clownfish. In terms of figuring out which genes are different between these colored tissues in the clownfish, we created the first transcriptome for the Amphiprion percula species, and we used that to carry out a differential expression analysis, which told us which genes are being used differently in each tissue. And then finally, in terms of what do these genes do, you might not be able to see this on the board. Oh, you can. We found that orange and white tissue weren't really conclusive in terms of our study, but the interesting finding and takeaway from this project was that immune function in black tissue seemed to be enriched, which might suggest black tissue has something to do with clownfish living in their anemone hosts. So that basically sums up my project. In the future, we're actually in the end stages of writing up a manuscript for this, so we hope to publish the information. But thank you all for listening. Are there any questions? Yeah, Liam.
So I know you're a biology student. What got you interested specifically in marine biology and this genealogy stuff?
Sure. So the question was how I got interested in this particular project. And what I have to say is I was looking for labs early on in my college career, and I was interested in animals since I was a really little kid. And so I was looking at labs that had to do with biology and ecology. And I got involved in the Boston lab, which helped me carry out this project later on. So it was kind of like I was already working with these fish, and the next step was to move on and do a project with them. Okay. Oh, Becca.
So you said the muscle finding in the orange tissue, there was no literature to back that up. Do you think there's something that you could investigate further and see if there's something there?
Sure. So the question was, we found that muscle genes were overrepresented in orange tissue, and right, we didn't find any literature findings that might suggest why that is, but that is a route we could take further down the road to look into why this orange coloration happens. There's actually not that much literature out there on why orange coloration happens, so it's something to investigate for sure. Thank you.